Canadian Bioinformatics Workshop, Module 2, Lab number 1. First of all, I just want to remark that this lecture or this recording is all part of the Creative Commons license and is freely available to share and redistribute as you like. Just make sure there's proper accreditations as mentioned in the licensing agreement that you can see over here. So as I said just before, we're going to be working on Module 2 and it's divided into two different parts. So the main goal of this module is to find overrepresented pathways in gene lists, and it's set up as a lab. And there are two different parts. This is part 1, which is going to discuss gProfiler, and the second part will be to discuss GSCA. So first things first, what are we aiming to learn from this lab? By the end of this lab, I hope that you will be able to run a simple enrichment tool like gProfiler using a gene list that we've supplied and understand the main parameters and the outputs that you'll get from this piece of software that we're going to be using. The results from this lab are going to actually follow through to some of the future labs that we're going to be looking at. So make sure that you do every single one of the labs in the order that we're presenting them so that you understand down the road in future labs how things fit together. So I mentioned just a second ago, lab number 2 for module number 2 actually consists of two different parts. And here I'm going to be going over part 1, which discusses gProfiler. Now, the two different softwares that we go over in this lab use different types of data. gProfiler, which is an online tool that we've mentioned previously, takes a thresholded list. So it's a small subset of a list. And there are going to be data sets that are going to encounter which don't actually score an entire set or the entire group of genes or proteins for your organism and will only actually generate a small subset. And that's what we call a thresholded list. And we're going to do a slightly different statistical test that is optimized for that type of list. So gProfiler actually uses the Fisher exact test. There are certain mechanisms in its interface where you can actually specify the background. So for example, if you're using a proteomics data set and your proteomics experiment only actually found 6,000 proteins, and you're looking at just the top set of those proteins that are differentially expressed, it's not the entire gene set or genome set for the organism you're looking at. It's not 20,000 genes that are associated with that genome, for example, for human, but actually it's just the 6,000. So your background actually can change. You've only actually identified 6,000 proteins. So there's a setting in gProfiler where you can go about setting the background as well. In addition to the Fisher's exact test, there's also the ability to use a minimum hypergeometric test. Now this one actually steps through your list. So if you have a ranked list of, let's say, 10 or 20 proteins, it will actually run the enrichment on just the first one, and then the first and the second, and then the first and the third, and it will give back the test that actually generated the best statistic for you. And it's a feature within gProfiler which you have to select that it's an ordered list. There is multiple hypothesis testing, as we discussed in the lecture, that is available in gProfiler. There are multiple options, and we'll specify as you go through the step-by-step instructions for gProfiler, which is the one that we prefer, but they're all there and you can use them all. Now, pathway databases, there are multiple pathway databases that are available that come with gProfiler. They're updated maybe on a quarterly basis. I'm not exactly sure how often they update them, but they are usually associated with the ensemble data releases. Usually when there's an ensemble release, gProfiler will go and update the databases it has associated with it because it uses ensemble to define those data sets. But you're not limited to that because within gProfiler, you also have the ability to upload your own pathway database or gene sets of choice. So that makes gProfiler a good choice for a thresholded type analysis because you're not restricted to their sets of data. Lastly, because it is based on ensemble, there are actually a large set of organisms that it supports. So you don't have to be limited to human or mouse, for example. And we're going to go over just the different outputs that they have. So this is just a table where you can kind of compare the two programs we're going to be going over in this module. A lot of them are similar. They have similar outputs. They have similar inputs, but the one thing that I really want to highlight is that gprofiler is an example of a tool that you use for a thresholded list, so a subset of your list. And GSCA is a very good example for something where you would use a non-thresholded list, using your entire set of ranked genes, whether or not they're significant or not. So the data that we're going to be using for this practical lab is actually mutation is from a paper entitled Mutational Landscape and Significance Across 12 Major Cancer Types. So this was an exome sequencing of multiple tumors across 12 different major cancer types, they detected point mutations and small insertions across all these different cancer types. I believe there was close to 4,000 individual cancers that they had the exome sequencing for. And then they narrowed it down. They were looking for common mutations across all of these different cancers. And in the end, they actually got a small subset of 127 significantly mutated genes that they want to associate as potential driver genes in these different cancers. Now, these genes are not found in every single one of these cancers. It's just across the entire very large data set, these genes were more mutated in these cancers, and they have an associated significance for them, but really it's just that these 127 genes are significantly mutated in cancer. So this isn't a ranked list in any sort of way. This is just these genes have a higher propensity of being mutated in cancer. And so it is a good example of a thresholded list that you can use for this type of analysis. So just briefly going over the gprofiler interface, and I'm just going to highlight a few things that you'll need to set as you go through the analysis, and that you can just understand what they are for, right? So on the left-hand side here, you're going to see that you can input your gene list. Now, technically, it's not limited to gene names, because gprofiler will try and do some mapping, and you'll see a little bit later that you can actually specify what type of identifier this is, but we tend to work with genes, and I think as biologists, we tend to use identifiers that we understand. So thankfully, gprofiler works with gene names. If there's something in there that it doesn't recognize, it will come up and tell you, listen, we found multiple mappings for this gene. Which one do you think is the best one to use? That actually often happens when you do a search with gprofiler. So you just have to look at the ones that it's giving you, and often it will be relating to ensemble annotations. And what I often do is select the one that has the most annotations, right? Because there could be multiple mappings for a given gene. And as I mentioned before, there is this concept of this minimum hypergeometric test where you're giving it an ordered list, and you can actually specify that it's an ordered query, meaning that your first gene in the list is actually the highest ranked, for instance, and it will actually go through your list, and it will take subsets of your list and do the exact same test over and over again and give you the best group of genes that give you the best best enrichment for a given pathway. It's not something we're going to be doing for this particular example, but I just wanted to point it out in the future if you do have yourself a situation where you have a ranked gene list and maybe you've assigned a threshold, but it's like a little bit of a fuzzy threshold. That's always the issue when you come to a threshold, is that you need to define that threshold, and sometimes we don't know whether or not it's a good threshold or not. So also I mentioned before the ability to set a background. So by default it's set to only annotated genes. So if you're looking at humans, it's basically using all human genes that are annotated. But if you have a data set where maybe you're not detecting all of human genes and there's a subset of genes that you are specifically detecting. So the example I gave before was proteomics. So, proteomics data sets tend to be a lot smaller in the amount of proteins that they can detect. So that's an example of where you might want to specify, no, wait a second, it's not the entire human annotated genes, it's actually the subset of proteins that I was able to detect. And then there's the ability also over here to indicate which type of multiple hypothesis testing you want to use. We recommend using the Benjamin E. Hochberger, but there are a few other ones that they have programmed into gprofiler that you can use. And then this is the threshold of gene sets that you want returned. So I don't have it highlighted here, but I'm just going to mention it as well. There is a setting here called all results. So by default, gprofiler is just going to give you the set of pathways and gene sets that pass this threshold that you've specified here, which is fine, right? You only want to see the significant results. When you have a data set that is not quite as strong as another data set, there are you might not get as many results coming back, and you might want to have the ability to play around with those thresholds a bit, and that's why we tend to recommend that you specify all results so that when we get to the later stages, we can see maybe pull out the pathways that are not coming up as significant, just to make sure that maybe they align with what we're looking at, because often we can have data sets that are pretty noisy, and it just might be it's a mechanism by which you can like investigate your results and maybe try and uncover what the issues potentially could be. And last but not least, over here, I don't have it expanded over here, but there's a set of this is where you can define what type of gene sets you want to use, go by a biological process or a set of pathway resources, or alternately, there's also a tab here where you can upload your own pathway and gene set definition files. So exploring a little bit of the results. So at the end of the day, after you've run your analysis, you're going to get to a screen like this. Obviously, follow the steps within the guided tutorial so that it'll get you there. It's a few steps before you actually get to this point. But over here, you have a data set or a pathway set that it's showing you results for. So if you're using multiple sources of data, gprofiler will divide it by each source. So this over here is Go molecular function. You have a list of each row represents a gene set or a pathway. So over here you have chromatin binding, DNA binding, and associated with each one of those pathways you have a p-value over here. This is the adjusted p-value, so it doesn't actually show you the original p-value. It shows you post-correction. And what you'll see over here at the other end, you'll see colored boxes. Now these colored boxes will only come up for Go gene ontology, so molecular function, cellular component, or biological process. And what these colorful boxes is actually indicating it's how this term was annotated, or how this given gene is associated with this term. Now, you'll get these boxes for every single one of your pathway data sets, but you won't get the colored boxes, because these colored boxes actually say how the annotation was found. So with Go or with gene ontology, there are different ways an annotation can be associated with a gene, right? So one of the examples is inferred from electronic annotation. So there are processes that will go through all of other species and will map an annotation from one species to another, and that's inferred from electronic annotation, so it's an inference. There are other really good terms, right? You have a curator who's read a paper and who has associated that annotation with that paper, so that's a type of annotation that's better than others. So just going to like highlight it. If you mouse over some of these, you'll actually be able to see what the annotation is coming from. Okay, so that's just a brief overview of what this lab is about. Please go ahead, go to Module 2, Lab Practical number 1, gprofiler, and start running your analysis. Now, after you've done that, there's also the ability to run gprofiler programmatically from R. So there are a few links at the bottom of the page if you actually want to try running gprofiler from R, but please do the step-by-step instructions with gprofiler before attempting to do it in R.